we're interested in developing aptamers that will be used on sensors. And so thinking about sensors and how one might optimize them to detect biomarkers. Of course, some of the characteristics that we want are, and we're looking at detecting viral infections, inflammatory and toxic agents, reproducible signal, high signal to noise, readable output. And then we're interested in real time. So patient side, on site, simple operation, no complex technical equipment, inexpensive. And finally, we want time resolution. That is wearable in situ. So for that, we need stable aptamers over at ambient temperatures in the presence of biological matrices and even at 37 degrees in some cases. So, of course, antibodies have been the go-to receptor for sensors for a long time, and they've worked very well for many assays and many means of detection, but they're not inexpensive. And so one benefit of aptamers then is that we can bring the price down, or expect that we should be able to bring the price down. So real-time aptamers are adaptable and inexpensive, but I think the real place where aptamers will shine will be in time resolution, because here we have molecules that can be much more stable than antibodies. It can be reused multiple times. You can watch the equilibrium changing as you move along, as long as you've got a stable molecule. And so the question is to have aptamers that are stable to the conditions that I defined here. And we've been working with 2'-fluoro-Y RNA aptamers, and I'm going to show you some results with those and how their stability will really contribute to our ability to make the kinds of sensors that we're looking for. So stability at ambient temperatures. Now, we did our studies in 50% serum at 22 degrees, and this compares a 2'-fluoro-Y RNA aptamer and the DNA equivalent sequence. And so, you're looking at days. Here we have the half-life. This is about 9 days for this particular DNA equivalent of this 2'-fluoro-Y RNA aptamer. We haven't gone out far enough yet to find the half-life of the 2'-fluoro-Y. If you extrapolate out, you say it's something about like 6 months to a year, but it's got to cross this line before we have a real value. And so we're starting these experiments again, but I won't be able to tell you the answer for probably a year or so. Now, that's not every aptamer. So a second 2'-fluoro-Y RNA aptamer is shown here. That's a mere 31-day half-life, but still significantly long for application to a wearable sensor. Another DNA aptamer also showed up here. You may not have noticed it, but similarly down to a half-life of about 8 to 10 days. So these 2'-fluoro-Y RNA aptamers are particularly stable under conditions that you might be using for a wearable sensor. As well, our other concern was stability if you have it in store it in a location that is not refrigerated and where you can, might be very humid, etc. So we also wanted to look at the question of, could we increase the stability of the DNA aptamer? Of course, you can modify the DNA aptamers, etc. But post-Celex modification brings with it the risk of losing affinity. And so our approach has been more make the aptamer, select the aptamer in the format you wanted, with the backbone you wanted, and move ahead with that. So, but here's stability to nucleases. So with DNA, as you can expect, if you add 1 millimolar DNA, EDTA, inhibit nucleases, extend the lifetime. Now, the 2' fluoro Y RNA, I always thought that that was inhibiting or preventing nuclease action, but actually, it's almost the same difference if you add EDTA to the 2' fluoro Y RNA aptamer. And so that was the one I showed on the previous slide, 31 days. We double the half-life by incubating with EDTA. So the main reason for the stability of the 2' fluoro Y RNA is probably not nuclease resistance, at least in serum, and it's probably structurally driven. So that was, for me, an interesting finding. So the other question that I mentioned earlier was stability to ambient conditions. What if you have it in the middle of Africa, and because we're looking at Ebola virus, and the packaging breaks, and you've got very high humidity, what happens to that sensor? And so we did some experiments where we had dehydrated aptamer. So basically, we just spun it down and dried it out, and we had a dried pellet and looked at the effect of humidity and high temperatures. And here we're looking at testing for a month at these conditions. So the untreated control was just left in the freezer, 23 degrees, 42 degrees, and then 42 degrees at 100% humidity. So basically take the test tube, put it in a container that contains water, screw the top, and get it to 100% humidity. And none of these treatments had any impact. So this is a binding isotherm. None of these treatments had any impact on the ability of that aptamer to bind. So that was good. Now, we did the same thing, and we're particularly interested in the 42 degree, 100% humidity with DNA. And for DNA, same treatment, and we're looking before and after, and well, it was there to begin with, but it is gone. It was totally gone after a month under these conditions. So again, the 2' fluoro Y RNA has become sort of our choice in terms of aptamers, but I'm going to talk to you about both 2' fluoro Y and DNA aptamers to the Ebola virus. So I'll start with the 2' fluoro Y RNA aptamer. We selected that to the Zaire, that is the Ebob Sgp. So Sgp is soluble glycoprotein. It is a protein that is a product of the same gene that the glycoprotein on the surface of the virus is. So if you're lucky, you'll get an aptamer that binds to both the virus and to the soluble glycoprotein. The nice thing about the soluble glycoprotein is because it's produced from the same gene, it tells you exactly the trajectory of the viral proliferation in the cells, and it's secreted immediately in the, once you have a full-blown viremia, you have 1 micromolar of this protein in the blood. So you'd have to have a pretty bad aptamer not to be able to detect that. And this, the ones that we've gotten have been in the range of 10 to 20 nanomolar KD. So this is the aptamer. It is specific for binding to the soluble glycoprotein over a number of proteins that predominate in the blood, including albumin. Now, we haven't gone up to the 600 micromolar yet, and so I can't say that once we get up there, we will be as effective, but we have done our studies in 10% serum, and so binding studies, not this one here, but I'll show others later on. So we know that we can detect in the presence of serum. We also asked the question, is this loop important for the aptamer to function? We made a truncated loop, and here you see that this doesn't bind, really, that's just a non-specific binding, which will goes to maximum. So the loop is definitely important for binding in this particular 2' fluoro Y RNA aptamer. Now, we also isolated DNA aptamers. Now, I'll say one thing about that RNA aptamer, and that is, or it's not RNA, 2' fluoro Y RNA, aptamer. And that is, it binds the soluble glycoprotein, but it doesn't seem to bind the GP1/2 on the surface of the viruses. So we were in parallel doing a similar selection with DNA aptamers, for DNA aptamers. And in this case, the one I'll show you does also bind to the viral surface. And so we, here you can see again, the binding to the Ebola virus. In this case, we only use 1% serum, but in the presence of 1% serum, to the Ebola virus Sgp. And this is the Sudan Sgp. And this is just an other protein, LCN2, which is actually NGAL. Now, the relationship between Zaire and Sudan is the following. So you can see that they're about the most different evolutionarily speaking over the evolution of this Ebola virus. So the likelihood is that although the KD may be different, that this aptamer will bind to all of the Ebola virus species. And that's been one of the problems with some of the antibodies, that they're only specific for the Ebob, the Zaire version. And here's another titration. In this case, we're looking at the question of human serum albumin. About 6 micromolar is as high as we can get before the assay just gives us results that are not meaningful, this particular assay, which is a filter capture assay. And so we can say that the Sgp aptamers are specific for Sgp. That is, they don't bind other proteins as to the extent that we have tested. And I always claim that specificity is only as far as you test it. But we have looked at a number of different alternative binding options, and we haven't seen that any yet non-specificity. Now, I'm going to get to sensors now, and non-specific adsorption to surfaces. Now, this is something that probably a number of you have come across, and if not, I'd be surprised. But because we're trying to develop sensors, we're also interested in this question of non-specific adsorption. And so we've developed an assay that involves using radiolabeled oligonucleotide and asking the question, to what extent, when we put that oligonucleotide on the surface, does it non-specifically adsorb, and can we remove that, and how much can we remove, et cetera. So we've sort of done some digging into the not so nice parts of making sensors. And so this test aptamer is the oligo I just showed you, the DNA binding, the oligo that binds to the Sgp protein. And this is what is predicted in a fold, M-fold, unifold, which it is now. And this is what we modeled as the potential three-dimensional structure. So what the data I'll show you relates to nanoporous alumina surface that is silanated with a compound referred to as Aptamers that has amino groups available, therefore, to do the chemistry to attach your aptamer to the surface. However, the data I'll show you is representative of data that we found with a gold surface, with polystyrene surfaces, with nitrocellulose surfaces. It is not specific to this particular one, but this is sort of where we've migrated to after having thrown gold out because it was so much non-specific binding, and discovering that that's probably wasn't our main problem. So this shows you the fraction of the total counts that we added to the surface that remained bound after washing. No chemistry here, just add the radiolabeled oligonucleotide and remove it, wash well with water, and here we have this result. Well, that's 53%. So the question is then, can we remove that? Is it responsive to ligand? Can we get it off this? So we first of all, by the way, tried various pretreatments, and we tried most of these actually with other surfaces as well. In fact, first with gold. None of these did anything beyond decreasing the amount of non-specific absorption by, let's say, 20%. And so not very effective. We want to get down to this range in terms of non-specific absorption. So then we tried treatments post-adsorption. And we thought, well, maybe the ligand will take it off. Well, it doesn't. Maybe heating it to 95 degrees for 10 minutes will take it off. No. And the reason we tried this one, the heating, was because we had also tried DNA phosphatase 6 molar urea. Nothing removes this stuff. So it is not only adsorbed, but it is irreversibly bound. Even the phosphate is not available for the phosphatase to remove, and that's of course our P32 label. So this did not look very good. And so this is, if you're using a sensor, I would guess that this is what's happening on your sensor. A lot of the material is just adsorbed non-specifically. So the question was then, how do we get around this? Now, I want to first of all show you some work we've been doing docking aptamers to the protein. And so this shows you 6011, and you can see that there's that stem area that I showed with a few bulges, and then this 3' region just doesn't seem to be interacting at all. By this model, remember that we don't have a physical structure of this, but we're modeling it. The 2' fluoro Y RNA aptamer, which is significantly smaller, this is 100 nucleotides. This one is 39. Seems to bind to about the same place on the protein. So we were interested in the question of this. Was this at all contributing to the non-specific absorption? Because actually our experience was that the 2' fluoro Y RNA aptamers had much lower percentages of non-specific absorption. So we take this structure again, and now we truncated it. And actually these two things were being done in parallel. We're now truncating it further, but to a 70 mer, and that 70 mer binds just as well as the 100 mer. So we call this 70 Sgp2A. We always give our aptamer, once it's less than what we've selected for, we give it a name. And you can tell how big it is by that name. So we did the same experiment. So here we have what was added, about a little bit over 10,000 counts per minute. Here's our 50% line with the entire. And now we get down to 20, and actually we've gotten down to about 12% binding of the reduced aptamer. So actually, the conclusion that I've come to is that the place you look first to solve your non-specific binding problems is the aptamer itself. And maybe that doesn't surprise some of you, but I, coming from a point of view of proteins, where with antibodies, your blocking agents are the proteins, and you just simply minimize the number of non-specific sites. That was sort of my first approach, and not very successful, as you saw. So then we wanted to know, what are the protein epitopes bound by these aptamers? And do they compete? Do the aptamers compete with each other? So we've got a 2' fluoro Y RNA aptamer, we've got a DNA aptamer, and you see that, so this is the radio-labeled aptamer. Here is the 6011. The radio-labeled aptamer here is a 39 Sgp1A, and they compete with each other. These are a few other aptamers that came up with 6011 and in other selections. Actually, this is a 2' fluoro Y RNA. They compete with each other. So here's the binding of 6011, and then any of these aptamers compete. Same thing if you turn it around. Here's 39 Sgp1A, competition. The thrombin aptamer doesn't compete. So it's a specific in that regard. So they compete on the protein for binding. What about with antibodies? So here we're looking at binding to 6011 and competition with 6011 with itself and another aptamer, SGP with when you have one of each of two antibodies present, no competition. So it's not binding. The aptamers are not binding to the same location as antibodies are binding. Now, so we can come to the conclusion here that the preferred binding site is different from the antibody. I think that's more generalizable. In fact, we heard a talk earlier where the same thing was observed, and I've seen that observed otherwise. So I think it may be quite generalizable that aptamers tend to find different locations than proteins, unless you actually sort of select for that option, at least the different locations than antibodies do. So the aptamer was predicted from what we've seen to bind at a different site. Now here's the PDB database information about the SGP protein. Actually, when they crystallized that, they used antibodies to crystallize it. So these are the antibodies, actually reduced antibodies. These are the FAB fragments that were used to stabilize the protein. So this is actually the protein, and it's a homodimer. You can see the two monomers. And when we modeled the aptamer bound to that, it bound at the interface, across the interface, and not where the antibodies were binding. So the modeling seems to be going in parallel with the biochemistry. Now, that's one of the questions we're asking. Can we get to the point where the modeling can turn around and tell us what we need to do next with our aptamer? We're not to that point, but we're doing a lot of comparisons to ask that question. So the aptamer binds across the interface. And so can we get biochemical evidence that says that it binds across the interface? And this it shows you the biochemical evidence for the 39-SGP1A. It's a disulfide bonded homodimer. And so dithiothiazole will break those bonds and you get a monomer. And we can demonstrate that on gels that we have now the monomer. This is a mobility shift assay. And so here we see the binding of the aptamer to the protein. Dithiothiazole eliminates that option. Thrombin aptamer has no impact. Another aptamer that binds to the protein competes with that binding, and so you don't see the binding. Similarly, with the 6011, different kind of assay, dithiothiazole doesn't have any effect on the basic binding, and here's binding to SGP, and here's the effect of dithiothiazole. So again, the modeling and the biochemistry match, which is always nice to see. So then I'm going to finish up by talking about some of our applications of these aptamers on nanoporous alumina to quantify SGP. Nanoporous alumina under the microscope looks like this. To your eyes, looks like that. That is, it's totally transparent because it's got these pores go straight through. So that actually in the middle there is the nanoporous alumina. And those are so we use membranes of nanoporous alumina that are placed in a sort of a laboratory rendition of a cell where everything's sort of connected by hand. Before I show that, this is the setup, working electrode, counter electrode, and two reference electrodes. And that gives the following result. Here we've done this in 10% serum or no serum, and there's really no effect of serum. Actually, sometimes the serum is advantageous, it seems. Added some HSA on top of the serum, but essentially we can see good binding in the presence or absence of serum. Now, SGP comes to 1 micromolar, and so if you dilute your serum by 10%, you're still way above the KD, which is about 10 nanomolar. So there's no problem in terms of being able to quantify this. And I should say also that when we put aptamers on surfaces, we have regularly found an apparent increase in KD and apparent increase in affinity. And so the KD goes down. So on the surface, this aptamer is about close to 1 micromolar KD, where 1 nanomolar KD, whereas it's about 10 nanomolar to 20 nanomolar in suspension. So usually we see a 5 to 10-fold increase in affinity on the surface. So that is with recombinant SGP. Then we have obtained so far only 1 sample, but a sample of serum from an infected macaque monkey. And that serum, when we did an ELISA, had a concentration of SGP in the 1 to 5 nanomolar range. And we found exactly the same result here. Now, what was done here was a standard curve was made by the recombinant SGP, and that's shown in the dashed lines here. The yellow shows the range of error in the standard curve, which obviously we still need to get that down, but these are the points of 2 different experiments with that sample, independent experiments showing that we can get exactly the same results with the sensor, or same values with the sensor as we can with an ELISA assay. And so conclusions, aptamer stability, very important for the kinds of things that we want, and 2' fluoro Y RNA aptamers are exceedingly stable. And so I think are a good option for real-time sensing wearable sensors. And they're less expensive than a lot of these highly modified aptamers where you have to make them chemically. We make them in the lab using the usual T7 polymerase, and we've also developed a way of adding any chemical moiety to the 5' end without having to get it synthesized. So, I mean, synthesized other than we do it. Applications to sensors, then non-specific absorption, I talked about that a bit, and I really have come to the conclusion, at least with our limited amount of data, that you've got to look at the aptamer if you really want to minimize your non-specific absorption. The aptamers, we isolated aptamers binding to the Ebola virus, SGP. They bind across the interface, different regions, and do antibodies, which might be an advantage at some point, although antibodies still have this stability issue that you can't apply in many cases. And we found conditions where we could sense the SGP with using impedance across nanopores. This is a label-free assay, so we're not using methylene blue or any other redox reagent. It's we're really looking at the redistribution. We believe we're looking at the redistribution of charge on the sensor, and so this makes it even more applicable to wearable and real-time sensing. And this is my group, and I have circled the individuals who have done a lot of the work here. The 3 people on the left did most of the work that you've seen. I cited them as we went along. Our collaborators, Monica Lamb is the computational scientist, Wendy Mori, virologist, and Pranav Kshatriya, the developing the sensors. And funding sources, as I mentioned in the beginning, Aptologic and also the NIH. And so I thank you for your attention, and I'm open to questions.